Alright, so in cell division, in mitosis at least, has 4 phases, and then cytokinesis. The longest phase of the cell cycle is interphase, which is divided into 3 parts. G1 is the first phase. In this phase, the cell grows and duplicates its organelles. The second phase is S, in which the cell copies its DNA for the final step, which is G2. In G2, the cell continues to grow and checks itself for any errors. If an error occurs, the cell will try to fix it, and if it cannot be fixed, it will essentially explode in a process called apoptosis. After interphase, we have prophase. During this phase, the cell membrane disappears and the chromosomes condense. They condense so nothing harms the DNA which the chromosomes hold during cell division. During this phase, spindle fibers also appear. They work to move, separate, and organize the chromosomes. Following prophase, there's metaphase. This is where the chromosomes line up for anaphase. The chromatids are separated and pulled to opposite ends of the cell. Finally, we arrive at telophase and cytokinesis. The nuclear membrane forms around chromatids and our helpful spindle fibers disappear. The membrane pinches together, but for plants, growth occurs in the cell plate to divide the cells. Now, at last we have our 2 identical daughter cells, including mitosis. Now, maybe some terms confused you if you're unfamiliar with mitosis. Here's a helpful clear-up. Chromosomes are structures of DNA that kind of look like thread. Cool, right? They also have protein in them. They allow cells to make exact copies of themselves. We briefly went over spindle fibers, but in simple terms, they are these structures of protein which aid in separation of chromosomes. And lastly for our notes, we have nuclear membranes. But first, what are these? Nuclear membranes are a layer that controls what goes in and out of a cell. It covers the nucleus, which is this round structure within a cell that houses DNA, or our chromosomes. Now, meiosis is relatively similar in terms of process, though meiosis is for sex cells, while mitosis is for body cells. We'll take our previous diagram for mitosis and then double it, minus the second interphase. This segment will be summed up easier to make this video shorter. In prophase 1 of meiosis, the spindle fibers form and homologous chromosomes pair up. Easy, right? Well, a new addition to this is also called crossing over. This is when the DNA gets mixed and matched so we don't look the exact same as our siblings. I'm going to skim through this next segment because it's relatively the same to mitosis. So for metaphase 1, the chromosomes are lined up to be pulled apart. In anaphase 1, now they're pulled apart. And in telophase 1, they separate and membranes form. Okay, this is the first set out of the way. In prophase 2, almost the exact same process occurs. The chromosomes condense again. You can kind of assume the rest. They line up in metaphase 2, separate in anaphase 2, and finally we conclude these phases with telophase and cytokinesis. These form the membrane, split the 2 cells in telophase, and now in cytokinesis we have 4 haploids, your full set.